All right, good evening, everyone. Welcome to the kickoff of the UCI School of Medicine Dean's Distinguished Lecture Series for the calendar year 2025. Happy New Year. I'm Dr. Michael Stamos, Dean of the School of Medicine here at UCI. The Dean's Distinguished Lecture Series was established in 2019 to bring together influential leaders from healthcare and academia to share their insights, experience, and anecdotes with our faculty, staff, students, and community members. The intention is that each year we will have one speaker representing advances in clinical medicine, one speaking to developments in clinical research, and one, like our speaker this evening, representing innovation in basic sciences. I'm honored to inform you that tonight's speaker is a world-renowned molecular biologist and physiologist. Her name is Dr. Nancy Carrasco, and she will share with us her insight, experience, and vision in her presentation, which is titled A Look into the Black Box, Uncovering the Remarkable Biology of an Elusive Protein. We'll ask for you to hold your questions till the end. We'll have microphones available so everyone can share in a nice, robust Q&A. Before we get started, please be aware that this lecture is being recorded. You can see up there the cameras. And the full recording will be available to anyone that wants to view it on our website by the end of the month. So without further ado, I'd like to invite Dr. Jeff Abbott, who was the School of Medicine's Vice Dean for Basic Research and the Senior Associate Dean of Academic Personnel and Professor of Physiology and Biophysics, to the podium to introduce our speaker. Thank you, Dr. Abbott.
Thank you very much, Dean Stamos. Welcome, everybody, to our Dean's Distinguished Lecture Series. It's my great pleasure to introduce you a brilliant scientist and an amazing person. Dr. Carrasco will be sharing with us tonight her presentation titled A Look into the Black Box, Uncovering the Remarkable Biology of an Elusive Protein. It promises to be a fascinating talk. Nancy obtained her MD and Master's in Biochemistry from the National Autonomous University of Mexico in her native Mexico City. Dr. Carrasco did her postdoc work at the Roche Institute of Molecular Biology in New Jersey, for which she received a Fogarty International Fellowship. Dr. Carrasco joined the faculty of Albert Einstein College of Medicine in 1987, where she cloned the sodium iodide symporter, NIS. That protein is going to become very important later. A breakthrough in thyroid pathophysiology with ramifications for many other fields, including structure function of transport proteins, molecular endocrinology, gene transfer studies, cancer, and public health. And she has served on the EPA's Science Advisory Board. Dr. Carrasco then joined the Department of Cellular and Molecular Physiology at the Yale School of Medicine in 2011, and in July 2019 became the chair of the Department of Molecular Physiology and Biophysics at the Vanderbilt University School of Medicine. Dr. Carrasco has received numerous scientific awards, including the Pew Award, the Arnold and Mabel Beckman Foundation Award, the Maria Sibylla Merian Award from Germany, the Merck Prize from the European Thyroid Association in Poland, the Rose Pitt Rivers Lectureship at the British Endocrine Society meeting in Scotland, the Noon Shavit Award, Israel, the Marshall S. Horwitz Faculty Prize for Research Excellence, the Light of Life Award, and the American Thyroid Association Valerie Ann Galton Distinguished Lectureship Award. She has served as president of the Society of Latin American Biophysicists, and Dr. Carrasco was elected to the National Academy of Sciences in the US in 2015, to the National Academy of Medicine in 2020, and to the American Academy of Arts and Sciences in 2022. She was named the Biophysical Society Fellow in 2023, and last February, she received the Biophysical Society Award in the Biophysics of Health and Disease. It is my distinct honor to welcome to the podium a remarkable researcher and great friend, Dr. Nancy Carrasco.
Thank you very much, Jeff, for that extremely kind introduction. It's an honor to be here. And what I would like to do today is I value the opportunity to tell you what inspired me to tackle this problem, where we are now, the unexpected directions in which it has led us and where we would like to go to. I've had a wonderfully interesting day talking to old friends and making new ones. And I'm looking forward to tomorrow as well. So the story actually started towards the end of the 19th century when Baumann discovered for the first time, a professor in Germany, that we accumulate iodide in our thyroids. And it was not known how or why. A few years later, Kendall at the Mayo Clinic started making extracts from the thyroid gland and realized that there was an important compound that, when injected into animals, increased metabolism, the basal metabolic rate. And he was determined to purify this compound. And he worked very hard for a long time. And the story goes that finally, on Christmas Day in 1914, he succeeded in precipitating the compound and isolating the structure that we see here, which is the thyroid hormone called thyroxine. So as you can see right away, the structure of this hormone is different from other hormones like glucagon or insulin that are made in the pancreas, and they're peptide hormones. Or we have hormones like estrogen, progesterone that have a cholesterol backbone. Thyroid hormone is very small. It's made by a couple of phenolic rings that are linked together by an ester bond. And the hormone is iodinated in 4 positions, giving rise to T4, or 3 positions when this iodine atom is not there. And T3 is actually more active. These small hormones are absolutely essential for the development and maturation of the central nervous system, skeletal muscle, lungs, and the intermediary metabolism of all cells. Now, what we have here is a condition that should not exist in the world, but unfortunately, it still exists. This is what happens under conditions of extreme iodide scarcity, iodide deficiency. These are adult women in Central Africa. They're standing next to a man of normal height, and they are physically impaired, cognitively impaired, and this is exclusively due to the lack of iodide in their diets. When I say that this shouldn't happen, it's because it's very easy to ensure that all of us have the appropriate requirements of iodide just by ensuring that we consume table salt that is supplemented with iodide. And guess how much this costs per year per person? A couple of cents. And it doesn't happen everywhere. We can see throughout history representations of goiter, what happens where there's iodide scarcity. And even as recently as 1996, this article was published on the first page of the New York Times, describing the gigantic problem of iodide deficiency that still happened in China. Fortunately, there was a very intense campaign to promote the use of iodide salt. And we can see the change that has occurred in the last few years. In the 90s, there were over 100 countries where there was different degrees of iodide deficiency. And now the situation is much better. So If you hear that some people think that one shouldn't consume salt that is supplemented with iodide, that's not a great idea. 50 years after Baumann discovered that we accumulate iodide in our thyroids, Saul Hertz, a very insightful physician in the Boston area, thought, well, if we have a system to transport iodide into the thyroid, we don't know what it is, but we may as well use it to transport radioiodide, radioactive iodide. And this was the beginning of nuclear medicine. So since 1946, patients with thyroid cancer have been treated very successfully by giving them radioactive iodide. And that's illustrated here. The cartoon represents a patient with a thyroid cancer. The treatment consists of removing his thyroid surgically. And then the problem, like in all cancers, what really poses the therapeutic challenge is the metastasis. So the white circle there represents a thyroid cancer metastasis to the lung, and the red circle, the iodide transporter. So as long as the metastasis still express the iodide transporter in red, then patients can be treated with radioiodide, and this treatment has been in place, as I said, since 1946. Now, this is no longer a cartoon. This is a whole body scan of a patient with widely metastasized thyroid cancer. The dark areas represent metastases, and we can see them literally when they are imaged with a low dose of radioiodide. And after a single dose of therapeutic dose, we see that the effect is quite noticeable. Some of the metastases are gone, and some are considerably smaller. So this therapy is still the most effective internal radiation therapy in place, and it's been used very successfully for over 75 years. It's quite specific, inexpensive, widely available, and importantly, the side effects are minimal compared to those of traditional chemotherapy. But we still didn't know the identity of the molecule that transports iodide. And believe it or not, there were endocrinology textbooks in the 80s still questioning whether the transporter would be a lipid or a protein. So when I became an independent investigator, I thought, this is very strange. Here we have a system that is used every day in clinical medicine in the evaluation, diagnosis, and treatment of thyroid disease, and we don't know anything about it. So we decided to go after it. And before that, I'd like to tell you a couple of things about the thyroid. This is a wonderful drawing made by Leonardo da Vinci in 1510. And it was not known what the role of the thyroid was. At one point, it was proposed that it was to separate the head from the rest of the thorax. But the thyroid is very unique. It's found here in front of the trachea. And if we take a closer look at the histology, it is organized in follicles. So you have to imagine this in 3 dimensions. So a follicle is made by a single layer of epithelial cells, and they're surrounding something that looks very homogeneous under the microscope called the colloid. The thyroid, the main function is to synthesize the thyroid hormones that contain iodine, as you know now, and it's the only hormone that is made both extracellularly and intracellularly, as we'll see in a second. And it has an enormous capacity of growth. There are patients with goiters who have goiters that are as big as their heads, as you can see here. So it's organized in follicles. And now if we have, this is a close-up of one single epithelial cell. And the strategy that we took was we obtained some highly functional thyroid cells, rat thyroid cells, a cell line that was derived in Italy by Ambesi in Piamboto. And when these cells are incubated in the presence of radioactive iodide and sodium, we can see that they transport iodide very nicely. If sodium is replaced with any other monovalent cation, there is no transport. And we can calculate the gradient. They accumulate about 100-fold. So we said, OK, clearly those cells express the transporter. We isolated the RNA, injected it into frog oocytes. These Xenopus levis that Jeff has been using for a long time in his lab, their abdomen is filled with oocytes. And these oocytes are big. They're like 1 millimeter in diameter. And if we inject mRNA, they make the proteins. So we took RNA from these cells, injected it, and asked, do they transport iodide? And they did, and in a sodium-dependent manner. So we said, OK, then we'll just make cDNA libraries and make transcripts, inject them, and we will find the piece of DNA coding for the transporter. So it sounded easy, right? Well, it took a very long time. But eventually, I won't get into the details, eventually we've found one day, with these experiments, we would see no signal whatsoever. And one day there was this minuscule, really small signal, but it was sodium dependent. And we thought, well, if the clone is there, then if we divide into smaller groups, we should get a larger signal. And that's exactly what happened. And we named the protein NIS. And NIS stands for natrium, sodium, iodide importer. As importer is a protein that transports simultaneously and in the same direction. So what NIS does is to take up iodide and transport it against its concentration gradient. It accumulates it. It's a process that requires energy. And the energy is provided by the downhill movement of sodium ions. So iodide, as I said, all the iodide that we need to make thyroid hormones comes from the diet. But we find it at very low concentrations in the extracellular milieu, in the blood, submicromolar, really small concentrations. So NIS had to evolve in such a way that would take iodide with very high affinity. So finally, we had the DNA that codes for it. And then NIS then mediates the first step in the biosynthesis of the thyroid hormones. I told you before that the thyroid hormones are very small. Well, to make them, the thyroid first has to make a gigantic protein, thyroglobulin. And thyroglobulin is made in the endoplasmic reticulum. And then eventually is released into the colloid right here. This big protein has many tyrosines. Tyrosin is the residue that gets iodinated. So NIS takes up iodide from the outside of the cell into the cell, then it gets into the colloid. It's oxidized and incorporated into a few tyrosyl residues. And we all have iodinated thyroglobulin at very high concentrations, over 100 mg per ml. Now, if you or I were to stop having any iodide in our diets, we would do OK for about 5 weeks or so, because this is an excellent reservoir. What happens is that when the concentrations of T3 and T4 go down, that triggers a negative feedback mechanism. TSH, thyroid stimulating hormone, is released from the pituitary. It binds to its receptor right here. and sends a signal so that this iodinated thyroglobulin is endocytosed back into the cell. Endocytic vesicles fuse with lysosomes and degrade all these gigantic molecules into pieces, and finally T3 and T4 are released into the bloodstream. So as you can see, this is a very convoluted mechanism. Several of the components had been identified at the molecular level, but not the iodide transporter. So finally, we got it. And then this is to show you once more the importance of the thyroid hormones. So if we, in a child that is making normal amounts of thyroid hormones, the developmental age correlates with the chronological age, as we can see here in this red line. However, when there are not enough thyroid hormones, the bone age, the height, or the mental age does not correlate, the development does not correlate with the chronological. Now, if we add thyroxine, if we give thyroxine, the thyroid hormone, you can see that there is improvement, but unfortunately, not when it comes to the mental age. So it is essential to, in many countries of the world, after a newborn is born, there is a test to make sure that they are synthesizing the right amounts of thyroid hormones. And if that's not the case, it's important to start giving treatment immediately. And the treatment consists simply in replacing what they are not making for whatever reason. Now, as you may already imagine, one of the reasons would be not having enough iodide, right? Or having mutations in this. Okay, so I told you that when Baumann was isolating the thyroid hormones for the first time, he was injecting them into animals and looking at their metabolism. So there is a correlation between the amount of thyroid hormones and the basal metabolic rate. If the amount of hormone is higher, patients develop hyperthyroidism. In contrast, if it's lower, hypothyroidism. And maybe some of you know people who have either hyper or hypo. These are common endocrine diseases. In hyperthyroidism, the metabolism is increased. So patients are nervous, and they sweat, and they tremble, and they have difficulty concentrating and sleeping. And in contrast, patients who have hypothyroidism tend to gain weight, and they are depressed, and they get fatigued. So going back to NIS, that mediates the first step in the biosynthesis of the thyroid hormones. Once we had the piece of DNA, we wanted to know what the protein looks like, right? So we sequenced the DNA in the lab, and as we were literally getting the sequence, we were translating it into protein and deriving the primary sequence. We proposed a secondary structure model, and then we experimentally tested. Without getting into the details, I'll tell you that NIS is a very hydrophobic protein. It traverses the plasma membrane 13 times. At that time, the dogma in the field was that transporters traverse the membrane 12 times. So the N-terminus and the beginning of the protein and the C-terminus, the end, would face intracellularly. That was the dogma in the field. But in science, we always have to test any ideas, and it became clear that in the case of NIS, it traverses the membrane 13 times, and the N-terminus, the beginning of the protein, faces the outside. And as we'll see later, this will become important. The red branches represent sites that are glycosylated, carbohydrate that is added to those sites. That's not essential for activity. And the most hydrophilic part of the protein is the C-terminus, which is phosphorylated. Now, I've already told you that the concentration of iodide is very low, it's submicromolar. It turns out that there are transporters and channels that transport chloride, another halogen, and they all transport iodide. But that becomes irrelevant, physiologically speaking, because we have 100 millimolar chloride surrounding the cell and sub-micromolar concentrations of iodide. But in the case of knees, it became clear that it has to discriminate in an exquisite manner between the two, otherwise we all would be hypothyroid, right? And we were very interested in trying to understand how does that happen. There are patients who are born with mutations in knees, and this is an autosomal recessive condition. If it's not diagnosed right after birth, then there's irreversible consequences, particularly related to cognitive issues. These mutations have actually been incredibly informative. The study has enabled us to discover remarkable properties of knees. So some of them are indicated there. They're single amino acid substitutions. They render the protein inactive, so that in and of itself is not that interesting, but then we try to understand what are the molecular requirements at those positions, and by doing that, we learned a great deal about how knees folds, how it travels to the plasma membrane, and how it transports sodium and iodide. And now we can place those mutations in the tertiary structure of the protein, and I will get to that later on. But I would like to talk for a little bit about an environmental pollutant, perchlorate. Some of you may have read about perchlorate because it's discussed every now and then in the general press. So the structure is here, is chloride with 4 oxygen, so it's an oxyanion, and it's used as solid rocket fuel. And it has been known for a long time that it inhibits thyroid iodide transport, okay? So we got very interested in the mechanism because unfortunately, the amount of perchlorate in the drinking water in many places, and actually California is one of those, Northern California particularly, and the amount of perchlorate in the drinking water increases, particularly close to military and industrial sites. So we thought, well, how does it inhibit? Does it block the protein somehow, or is it transported by knees? And this would be a trivial question to answer if we had access to radioactive perchlorate. We do transport assays with radioiodide all the time. But we don't have access to radioactive perchlorate, so we relied on electrophysiology. Remember that the stoichiometry of transport is 2 to 1, 2 sodiums per iodide, so the process is electrogenic. So if we do electrophysiological experiments in oocytes, for instance, and the oocyte is perfused with a solution that has sodium, the moment that we add iodide, we see these currents that represent positive charge going into the oocyte, because the stoichiometry is 2 sodiums per iodide. When we did the same experiment with perchlorate, no currents. So that's a negative result. There are many possibilities. And we were intrigued by that, but the question became more pressing because NIS was supposed to be a thyroid-specific protein, only expressed in the thyroid. And then we and others discovered that it is actually expressed in other tissues. In the salivary glands, for instance, so the concentration of iodide in the saliva is higher than in the bloodstream. It is expressed in the stomach, basolaterally also, so the concentration of iodide in the lumen of the stomach is higher. In the small intestine, there it is expressed apically, because that's how we absorb iodide, all dietary iodide. And very interestingly, it is expressed in the lactating breast, but not in the non-lactating. And if we think about it, it makes a lot of sense, because what's the source of iodide for the newborn? What is found in the maternal milk. And as I've said over and over, right after birth is absolutely essential to synthesize the right amounts of thyroid hormone, but not expressed in the non-lactating. So then we said, so what happens under other conditions where there's a lot of mitogenesis, when the cells are replicating, and that's thyroid cancer, right? And to our surprise, this is expressed in a high percentage of breast cancers, regardless of whether they are estrogen and progesterone receptor positive or negative, HER2 negative positive. But then what we really wanted to understand is, are they expressed in the metastasis? Because after all, that's what poses the therapeutic challenge. And this is functionally expressed in the metastasis. Now, this is a figure that comes from one of our collaborative papers with Jeff. And I like it very much because it shows very clearly how iodide is accumulated in all the pairs of lactating mammary glands. So if NIS is expressed in other tissues, and perchlorate is going to affect transport in those tissues, that's even more relevant. So the answer came actually from experiments that did not require super high tech. So that's satisfying sometimes that one can obtain an answer without super high tech. And basically, we just place cells on a filter so they would be polarized. And by doing that, we showed that perchlorate is actively transported by NIS. So first in cells. Then we showed that it's transported and accumulated in the milk in rats. And when we published those results at the beginning, we actually got a lot of heat in the press, particularly from those industries that contaminate the water with perchlorate. And our findings were replicated by other labs. And we were delighted when the Obama administration announced for the first time in 2011 that there was a plan to start regulating the amounts of perchlorate in the drinking water. So we were delighted, and then we were appalled when the Trump administration decided that they were not going to regulate the levels of perchlorate in the drinking water. And we're talking about something that interferes with cognitive development. So I think I don't need to say more. But Alex Llorente, a very talented grad student in the lab, really wanted to understand the mechanism. I won't get into the details now, but it turns out that, and together with Mario Ansel and Jeff, we showed that perchlorate not only is transported by NIS, but even very low concentrations of perchlorate change the mechanism by which NIS transports iodide. So the driving force changes. The process is no longer electrogenic, so there's less transport. It decreases the affinity for iodide. And of course, the most sensitive populations are those that require the exact amount of thyroid hormone. So pregnant, fetuses and infants, and of course, pregnant women and nursing women, because during those stages of life, the requirements for thyroid hormones increase. So science dubbed this the double punch of perchlorate. On one hand, it competes with iodide, and on the other hand, it changes the mechanism by which NIS transports iodide. I mentioned before that we have learned a great deal from the mutations found in patients, and I would like to share with you a couple of examples. These are not the patients. These are the students and postdocs in the lab who have studied them thoroughly. So, okay. When Vicky Paroder was a student in the lab, she had characterized a mutation that I'll tell you about in a second. And then we received a phone call from a hospital in Long Island saying that they had just seen a girl, an 8-year-old girl, with goiter, as you can see, with a stature of a 3-year-old, so really short. And we started discussing the case, and we said, well, have you done an imaging study? And when her thyroid was imaged, there was very little accumulation of iodide. It was not nothing, but it was very, very small. So we thought, well, maybe there's another mutation. And when her DNA was sequenced, she had a substitution, a histidine instead of an arginine at this position. So Vicky was very, we were all excited. Oh, this is going to be a new mutation. It was the same one. And she had just shown that in the case of this mutation, this is what NIS looks like when you run a Western blot. This is the mature part of the fully glycosylated protein, and this is the partially glycosylated when it's going from the ER towards the plasma membrane. And as you can see, in the case of the mutant, it never fully matures. It's retained intracellularly, and that's the problem. But then it turned out that the patient was not homozygote for that mutation. And I told you that this is an autosomal recessive condition. So something else had to be going on. And then we found that she had another mutation right here, a glutamate instead of a valine. And I also told you that when the imaging study was done, there was a little bit of accumulation. So we made the construct in vitro, transfected cells, and did a transport assay. These are cells that were not transfected. When they are transfected, when they're expressed in the wild-type protein, there is a lot of iodide transport inhibited by perchlorate. And with the mutation, there's little, but that's real. If we put another negatively charged residue, we see the same, but if we neutralize it, we recover some of the activity. And by Western blots, we showed that the protein, there's a little bit of protein that is maturing, consistent with the fact that there was a bit of accumulation. So then we did the kinetic analysis, and even this protein, the small amount of protein that gets to the membrane, has the same Km, the same affinity for iodide as the wild type. So we thought, well, could it be that what is retained intracellularly is as active, and that the only problem is that it is not reaching the plasma membrane? We made membrane vesicles from intracellular organelles, from inside the cell, and that's indeed what happens. So it's one of these cases where the protein gets retained intracellularly, but it's as active as wild type. And at that time, we didn't have the structure of NIS, but by homology model, we thought that the problem was that if we look at NIS from like inside the cell, all the blue areas represent positively charged residues, and this mutation introduces a negative charge. Now, the most satisfying part of the story was that the girl started receiving thyroid hormones, and she started growing beautifully, and the small amounts of thyroid hormones that she was making seemed to be enough not to affect her cognitive abilities. She's doing fine at school. Now, don't think that I'm going to tell you about every single case, but each one has been a fascinating story. So when Mia Reed was a grad student in the lab, she studied this mutation, B59E, but we were very intrigued by the fact that this was identified in Japan, and it turns out that there were two kids. I'm simplifying the story a bit. And both of them were unlucky enough to inherit both alleles from the parents. But the clinical manifestations were very different. The older kid was slightly affected, and the younger kid was horribly affected with the same mutation. So this didn't make sense. So we had to do a little bit of detective work. And it turned out that the mother was addicted to seaweed, to laminaria that contains enormous amounts of iodide. So we thought, and not only that, she breastfed the first kid, but not the second one. So that led us to hypothesize that because she was consuming astronomical amounts of iodide, she was passing that iodide into her maternal milk, and that the first kid, even though she has the mutation, was receiving these enormous amounts of iodide, and somehow iodide was getting into the thyroid. The second kid, unfortunately, was not breastfed and was horribly affected. So in order to test that hypothesis, we decided to generate the NIS knockout mouse. So, and this is an imaging study done, it's a SPECT-CT done with pertechnetate. Pertechnetate is another oxyanion, just like perchlorate, but it's used in clinical medicine all the time because it has a shorter half-life than radioiodide and is less expensive. So if we take a look at a wild-type mouse, as expected, it accumulates pertechnetate in those tissues that express NIS, in the salivary glands, in the thyroid, in the stomach, and it's eventually eliminated by the kidneys. In contrast, the knockout mouse does not accumulate any pertechnetate because it doesn't express NIS. These are just fiduciary marks. So we expected this mouse to be severely hypothyroid, right? It doesn't have NIS, it cannot make thyroid hormones. And when Rachel Kaspari and Fernando Ferrandino started analyzing these mice, they don't make a lot of T4. So in purple, we have the knockout, wild type and knockout, but they were making considerable amounts of T3. This didn't make sense. How are they making the hormone if they don't have NIS? So again, we had to do some investigative work. It turns out that the chow diet that we feed our mice with contains 40 times the daily requirements of iodide for a mouse. So in a way, we were recapitulating the story of the mother that was addicted to laminaria. So we then just fed these mice with a diet that contains 1x, not 40 times more iodide. And then they practically don't make thyroid hormones. And TSH, remember that there's a negative feedback mechanism, goes through the roof. So for the first time, so we were very excited because we realized that we have an animal model of hypothyroidism that we can regulate a priori depending on how much iodide we provide to the animals. And we have learned that severe hypothyroidism is not just an aggravated version of mild hypothyroidism. Without getting into the details, we have found some really interesting metabolic effects. For instance, severely hypothyroid mice are protected against non-alcoholic fatty liver disease. And there's a paradoxical effect. These mice don't gain weight. And this is due to the fact that they increase their skeletal muscle thermogenesis. And how do they do that? Well, they express this protein, polypeptide, really sarcolipin, which is a very small peptide that interacts with the calcium ATPase that is expressed in the endoplasmic reticulum or sarcoplasmic reticulum. So actually, the first two crystal structures of SERCA, sarcolipin was bound to it. And what it does, it uncouples SERCA. So when calcium concentrations increase in the muscle, that, of course, generates contraction and many other things. But eventually, those concentrations of calcium have to be brought back to the submicromolar levels. And that's done in part by SERCA. It takes calcium and transports it against its concentration gradient towards the interior of the sarcoplasmic reticulum. It's an ATPase, so the driving force is provided by hydrolysis of ATP. But when sarcolipin is there, it uncouples the protein. And what I mean by that is that it continues hydrolyzing ATP without moving calcium, or much less, and this generates heat. And so this incredible expression of sarcolipin, so more ATP has to be generated if this is happening, and indeed beta oxidation is increased. More recently, Paola Torres Manso, senior member of the lab, decided to feed these animals with a high-fat diet, and as expected, the wild-type animals become really obese, but the knockouts don't gain any weight. And one could think, well, maybe you're feeding them with a high-fat diet, but they're not absorbing the fat. That could be a possibility, but that's not the case. When we do bomb calorimetry of their feces, they eat as much as the wild-type mice, and their adipocytes don't expand. They don't accumulate triglycerides as the wild-type do in their livers, and when we analyze histologically their liver, the wild-type mice on the high-fat diet, one doesn't need to be a pathologist to see that there's hepatosteatosis, there's accumulation of lipid, but the NIS knockout mice are completely protected. So Paola is working hard in trying to understand the mechanism by which thyroid hormones upregulate, so low thyroid hormones, right? Because this was in the knockout animal. It's a negative regulation of sarcolipin, and recently she has developed an in vitro system, and here we can see that if we add thyroid hormones, the expression of sarcolipin goes down. Now we want to understand how does this cell autonomous effect occur mechanistically. And as I told you before, we really wanted to understand how does NIS discriminate between chloride and iodide. So Silvia Ravera engineered a construct, a NIS construct with a purification tag. We transfected this into used an lentiviral vector to express it into HEK cells, select for those cells that are expressing the protein, showed that those tags don't interfere with activity, and then purified the protein to homogeneity. So we were very excited. We thought, okay, we're ready to start some structural studies. And Silvia set up many crystallization trays with all kinds of conditions to no avail. And eventually we would get microcrystals, but they wouldn't diffract well. And this part of the story reminds me of Samuel Beckett when he said, ever tried, ever failed, no matter. Try again, fail better. Silvia was not deterred, and at that time we thought, well, maybe we should switch into cryo-EM. But the size of the protein of NIS, about 60,000, was considered small to be able to determine its structure by cryo-EM. So we were in the midst of trying to enlarge it by making all kinds of different modifications. And one day, Silvia became impatient. I don't know whom she got it from. And she just put the protein in the grids. And without getting into details, she collected over 1.5 million particles, and we got the structure by cryo-EM. And here I just want to show you how hydrophobic the protein is. It's completely embedded in the detergent micelle. And the APO structure, the one without any substrates, is open to the inside. For these transporters to work, they have to be open to the outside first, bind the substrates, they go through an occluded conformation, and then open to the inside. So the next thing was to purify the protein in the presence of sodium and iodide. And we were delighted when we saw 3 new densities, and we could assign those densities to the 2 sodium ions and iodide. So we trapped this molecule in the occluded conformation, so we can see exactly how NIS recognizes its substrates. And the surprise was that NIS recognizes iodide, here is the purple sphere, as a hydrophobic anion, which was quite a surprise. So most of the interactions occur through side chains of hydrophobic amino acids. And the 2 sodium ions are there, and I can talk much more about this in the lecture tomorrow. And of course, then we question, are these residues really essential for activity? We made a lot of substitutions, they all are. And all of these residues are 100% conserved from fish to humans. As you may imagine, the next question was, well, what happens in the presence of perrhenate? And we got the structure in the presence of the oxygen ion, and now there's only one sodium ion there. So if we put all these structures together, what happens is, and so these are occluded, and I told you that the APO structure was open to the inside. So the only thing that is different is that it's one residue, this phenylalanine, that blocks the exit pathway, and when the substrates are there, it opens it. So after we cloned NIS, we and others thought, wouldn't it be wonderful to introduce NIS into cancers that don't express it and be able to treat those patients with radioiodide, since the treatment has been so successful? And many groups have published papers of preclinical studies showing that this is possible. So if you introduce it into several cancers, treat with radioiodide, it has a profound effect on destroying those cells. The problem is that we want to protect the thyroid of the patient if we do that. So for a long time, we thought, wouldn't it be great to engineer a nice molecule that transports perrhenate? It turns out that radioactive perrhenate is more energetic than radioiodide, that doesn't transport iodide, and then we could protect the thyroid of the patient just giving cold, non-radioactive iodide. And this was like a dream for a long time, but having the structure and allow us, and also from information from another mutation found in a patient, that allowed us to concentrate on this region of the protein, that the alpha helix transitions into a pi helix. We made a lot of mutants, and without giving you all the details, we engineered a double mutant. And this double mutant hardly transports any iodide as compared to the wild type, but it transports perrhenate just as well. Not only that, when we look at perrhenate transport as a function of sodium, the wild type, as I've been saying over and over, is electroneutral. We get this hyperbolic curve. But we notice that with the double mutant, now it's a sigmoidal, highly suggesting that there's cooperativity, and therefore that the stoichiometry had changed from electroneutral to electrogenic. And this is eminently testable, particularly if you have a close friend who loves to do electrophysiological experiments, or people in his lab like Rian, who's somewhere here, who does the experiments. And with the wild-type protein, we see currents with iodide, but not with the PF mutant, the double mutant. No currents with perrhenate, but beautiful currents with perrhenate. So indeed, the stoichiometry, not only does it transport perrhenate, but with a different stoichiometry. And the next step then is it possible then to introduce this mutant into cancer cells and see what happens with perrhenate, even in the presence of iodide? This is just to show you by MD simulations that we have an idea why this happens. And basically, even with the wild-type protein, as we increase the concentrations of iodide, there's less perrhenate because they compete, or perternate, they compete. But even at 100 micromolar iodide, which is a huge concentration, there's practically no competition. So then we went ahead and treated cells for a couple of hours with radioactive perrhenate under these conditions. With the wild-type protein, over 75% of the cells were killed. But Alex and Hashwit are laughing here. They're very happy because with the double mutant, there's no difference. So the degree of destruction was the same, even in the presence of iodide. So we're very excited about this and doing animal experiments, and hopefully we'll be able to extend the role, the clinical role of NIS beyond thyroid disease. So a few years ago, we didn't know anything about the iodide transporter. At some point, it was called an iodide pump. People thought that it might be the same sodium-potassium ATPase that transported iodide, because when it was inhibited, of course, the sodium gradient is dissipated and there was no transport. Now, we think that this is a very cool protein in the middle of many things. And whatever we learned in these realms, they help us understand the other ones. So the more we understand this at a fundamental mechanistic level, the more we will be in a position to extend its clinical role. It's already being used as an imaging agent, for instance, in CAR T-cell therapy. And in the area of public health, we hope that iodide deficiency disorders will be eliminated, the perchlorate pollution of the water, and when there's nuclear fallout, iodide given right away, cold iodide can prevent future damage of the thyroid. And actually, when the war in Ukraine started, I got telephone calls, people asking whether they should take pills containing high amounts of iodide. Of course, I would not be standing here were it not for the contribution of many young and talented people who've been passed through the lab from many different countries. I think that science is a truly international endeavor. And these are the current members of the lab. We had a long and productive collaboration, beautiful collaboration with the late Mario Amsel, who was the head of biophysics at Hopkins, who sadly passed a couple of years ago. Now we continue collaborating with Mario Bianchet, a senior member of his lab. All of you know this person. We've had very unexpected findings, and we have some ideas of, again, how to modify the behavior of these proteins. And I think even someone who loves channels now is convinced that transporters are also important. And Erkan Caracas is a member of my department, a structural biologist, who taught us how to analyze all the cryo-EM data. And with Fred Seward, we assigned the densities of the ions. To end, I'd like to show this drawing that my son Eric made when he was very young. And as we were sequencing this and we were making our models of the primary sequence, he made this drawing. And I think that I'm afraid he heard a lot of NIST talk over dinner. But I was very pleased when once, right after we cloned this in Germany, when I was, they started a new institute of gender studies, and I was lucky enough I was invited to get the first award, the Maria Sibila Award. Maria Sibila was a wonderful artist and botanist in the 17th or 18th century in Germany. She decided to go on her own with her daughter to Suriname and make beautiful drawings that now are some stamps in Germany. And I had to give a talk at a museum, and the head of the museum told me, well, maybe you're a good scientist, but have you realized that your son is really talented? So thank you very much for your attention, and I'll be happy to take any questions.
Thank you, Dr. Carrasco. Fascinating look at the journey into the black box. So 35 alumni you have listed there, I think, right? 35, looks like 35. And so you said they were young. So how many years was that passageway? Some of them may not be as young anymore. How many years have we?
Time passes. Time passes, but we published the cloning of NIS in '96.
Okay, excellent.
And the structure.
Good perspective. Yes, thank
Yeah.
you. We have time just for a couple of questions from the audience. Any questions from the audience? Jeff, you can't ask the first one because you're a plant.
All right. I have two questions. I have many more questions, but I will ask only two. So in your experiment with iodine transport and perchloride, why wouldn't iodine oxidize by perchloride?
Because perchlorate is actually not such a strong oxidizing agent. And actually, what it does right away is that very, very low concentrations of perchlorate. Our hypothesis is that it interacts at an allosteric site in the protein that prevents one of the sodium ions from binding. So even transport of iodide is electroneutral.
Yeah, but typically perchloride can be used to oxidize iodine in test tubes. So did you check whether periodine was formed?
No, it's not formed. Right, so we don't have any evidence that it is being oxidized because we can do experiments where we can take that iodide like from the other compartment when we have polarized cells and see that it is transported fine. And if it were oxidized, it wouldn't be transported well by this.
Just one very quick technical question. So your structure shows the sodium that occupies those two sites, and sodium is 23 atomic mass, right?
Yes.
And that's not different than water. And with that resolution of the membrane segment, are you sure that those are sodium and not water? Particularly that sodium in your reaction required about 100 millimolar concentration?
Well, the KD for sodium is about 35 millimolar or so.
Millimolar, very high.
It's millimolar. So we don't have a structure of 1.6 angstroms resolution. That's what we could need. But we did experiments to replace all those residues and look at the effect on iodide transport and how the sigmoidicity changes and the affinity, apparent affinity for sodium. So it's very likely that that's where sodium ions are. In one case, the distance is within 2.5, 2.7 angstroms, so it's an appropriate distance to coordinate sodium. The other case is longer distances, but we have a cation-pi interaction. So we feel that it's very likely that those densities represent the sodium ions. And it's really interesting how when we have perrhenate, I didn't have time to get into the details, but there is a glutamine that is essential. It actually interacts with iodide through the gamma carbon and with the two sodium ions. And when we have perrhenate, it switches the conformation and it can no longer interact with one of the sodium ions. Now we have made a mutant where we don't have that glutamine, but we have an alanine. The protein doesn't transport iodide, it still transports perrhenate, and now we just got the structure, and sodium is being coordinated by other residues. So that's where we are.
Thank you. We have time for one more question, then we'll head out to the lobby for refreshments after we have a chance to give Dr. Carrasco a small award.
There's a question here.
Yes.
Sorry. I also have a question up here. To your right.
Oh, yes.
Yeah. Thank you so much for the beautiful presentation. I have a question about the perrhenate when you did the double mutation so that it exclusively transports perrhenate. With that being an oxyanion and perchlorate also being an oxyanion, does that introduce any conflicts with people that would get that treatment in a polluted area with perchlorate?
Well, we hope that the concentrations of perchlorate are not that high at the level that, if people drink water contaminated with perchlorate, they would have such a high concentration. Theoretically, yes, if they had, but there are ways to ensure that they wouldn't be drinking water that is contaminated with perchlorate. But you're right in the sense that perchlorate needs transports both in a very similar fashion, perchlorate and perrhenate. And actually, the affinity is higher than for iodide.
Got it. Thank you.
But we'll have time for we have one more question here. Go ahead, please.
So I want to know why specifically did you choose like a frog to clone cDNA in instead of any other organism?
A what? I'm sorry.
The frog.
The-
I want to know why you chose specifically the frog.
Okay, we didn't clone the cDNA from the frog. We used the oocytes because it's a wonderful way to express proteins. So when you inject RNA from other species, you inject the RNA and they then make the proteins and they carry out post-translational modifications. So it was a good system to screen many libraries. But the clone that we got was from rat. Now, transporters are among the most conserved proteins. So actually, human knees and rat knees are almost identical. There are there's an insertion of 4 residues and a couple of modifications, but they're very similar.
Excellent. Thank you all. Thanks for the great questions. Thank you, Dr. Carrasco, for a wonderful lecture. We have a little award for you. It says UCI School of Medicine, Dean's Distinguished Lecturer, Nancy Carrasco, MD, January 16th and 17th. Oh, it's quite heavy. Be careful.
Thank you very much.
Thank you.